Hi, thanks for the introduction. So today I'm going to talk about HealthSense, which is our platform for building software-defined mobile-based clinical trials. Since the advent of smartphones and wearable technologies, thousands of popular news articles have been written that claim that these technologies are the future of healthcare and will revolutionize healthcare. Unfortunately, through the prescription of mobile applications, mobile health applications. Unfortunately, this process has been somewhat slow. In order to understand why, we have to step into the shoes of a clinical researcher. So the job of a clinical researcher is to first develop a hypothesis. This hypothesis might be that physical activity leads to decreased symptoms of depression and anxiety, or that grief counseling can lead to better heart health. After a hypothesis is developed, the clinical researcher has to implement the trial. This consists of defining an objective, designing a study, building an infrastructure, and recruiting participants. After the trial is implemented, results are gathered, the trial is executed and results are gathered, and it's either determined that this intervention was a success, which leads to clinical adoption, or that the hypothesis is rejected, in which case the researcher has to go back to developing another hypothesis. Unfortunately for the clinical researcher, hypothesis rejection is more common than success, and as a result, researchers spend a while iterating through this loop of developing a hypothesis, implementing the trial, and gathering results. It's also important to note that in addition to infinitely many hypotheses, there's also many different types of trial designs. So association trials are trials where you have multiple streams of input data and you're trying to find correlations between observations. Randomized control trials are where you have multiple groups, each receiving a unique intervention, and you're trying to estimate the causal effect of that intervention. For N-of-1 trials, similarly, you're trying to prove causal effect of an intervention, but we only have one patient this time. So that single patient is assigned a random sequence of interventions. One of the interventions is usually a control intervention, and you're trying to determine the average causal effect. SMART is a trial technique that's used to determine causality in multi-stage trials, and micro-randomized trials are a way of determining causality for just-in-time interventions. Just-in-time interventions are a way to schedule interventions in a way that's optimal for impacting behavior. So an example would be if you're a clinical researcher and you want to encourage smoking cessation, you might create an application that when it notices a person is approaching a smoke shop, they are notified of the harmful effects of smoking. So back to our clinical researcher, as they're iterating through this loop of developing a hypothesis, implementing the trial, gaining results, and then going back to developing another hypothesis, the hardest part of this loop is building the infrastructure. To understand why, we have to take a look at what technologies are currently available to clinical researchers. So we can think of all available technologies on a graph of customizability by configuration simplicity. In the lower left-hand corner, you have single-use applications. These are applications that are built for a single hypothesis and a single trial design type. At the lower right-hand corner, we have some recording technologies. These are high in configuration simplicity because they're built by established tech companies with very easy-to-use interfaces, but they're low in customizability because they can only be used for association trials. In the upper left-hand corner, we have high customizability tools and frameworks with low configuration simplicity. So you can use these tools and frameworks to build any sort of trial design type or hypothesis, but they require advanced technical knowledge. So what clinical researchers need is a platform that is high in configuration simplicity, but also very customizable in that it can implement any trial design type. So our primary goals in developing this platform were to first make it possible to compose any set of sensor streams or survey questions in order to guide these interventions. We also wanted our platform to be highly expressible in that you can express any hypothesis or clinical trial design type. And we also want to build a platform that's simple and easy to use, such that clinical researchers with no advanced technical knowledge can use our platform. So first I'm going to go through how we approached composability and expressibility. We did so by designing a general trial design. This general trial design subsumes all other trial designs. So every other association, RCT, SMART, they're all special cases of this trial design. The generalized trial design consists of two main components. The first component is a starting initial state, which contains a list of participants, response data, and app configurations, and also a function that maps a state at time t to a study at time t plus 1. So I'm going to step through just 2 examples of how these other trial design types are special cases of this generalized trial design. So a randomized control trial starts with just a list of participants, each with their app configurations and response data. And then the first step is to divide participants into research groups. The first group might be a control group, and the second one is the intervention group. And then unique interventions are continually given to those groups, and at the end of the study, the results are analyzed. And here's an example of how just-in-time intervention is a special case of our generalized trial type. Again, you start off with the list of participants, each with their own response data and app configuration. You record internal and external sensors, as well as survey responses from each application. And then at each time step, you process the data and check a condition. If the condition holds true, then you give an intervention. Otherwise, you continue to record from sensors. So just to make this a little more concrete, every trial can be thought of as an initial starting state and some combination of features, such as sending notifications, enabling or disabling sensors, sending or using remote computational pipelines such as AWS Lambda, or accessing external APIs, third-party APIs like Google Places. And then all of this gets fed back into a new study state, which represents the app configuration response data of the participant. So the physical implementation of this is that all of this is somewhere on a remote server, and it's taking in a bunch of streams of data, and it's executing any sort of combination of these features that's consistent with the trial design criteria, and it's spitting out a new study. So how do we represent this generalized trial design, or how do we represent the process of creating this generalized trial design in a simple and easy-to-use way for the clinical researcher? So this is what the graphical user interface looks like for the clinical researcher. They start off by creating the study with the study metadata, including a study image, title, a description, and then they are prompted to input what sensors they want to activate and certain properties of those sensors. So if you want to record from location, phone usage statistics, app data, etc., you can select those to record during your study, and you can also set properties like a sampling rate and sample type. The researcher then adds questions to their question bank. These questions can be multiple choice questions, slider questions, image capture, food diary, etc. And then finally, they can set up a schedule, which this is where the function in the generalized study type comes in. They can set up their trial to be a randomized control trial through a graphical user interface, or a association trial, or any of the given trial design types. And finally, they can set up a list of participants, and this is a fully hosted recruiting service. So if you input a participant, they'll receive a notification with instructions on how to set up the app and an entry code. So what does this look like for the research participant? Well, the research participant starts by logging into their application. They can also view their to-do list. The to-do list has all of the survey questions that they need to answer, and it also has instructions on how to connect external for internal sensors. On 3 clinical trials. I'm only going to explain 2 of them here today because of the mobility trial. The goal of this was to see how mental health conditions correlate with in-person sociability and mobility. And so we had 3 groups of participants. The first group had psychosis, the second group had depression, and the third group was a control group. And we sent daily EMA questions, including questions like, how is your overall mood today? We also asked them to sketch a perceived sociogram. This is their daily connections, who they talk to, etc. We also recorded from a lot of different background sensors, such as call logs, SMS logs, GPS, and step counting. And then we asked them to connect HealthSense to an audio recording smartwatch, which recorded audio throughout the day, and then we downloaded the results. Our results were that patients with schizophrenia, or with psychosis and with depression had lower overall mobility and sociability than the control patients. And the second trial that we implemented was a contextual food diary. So we wanted to know, how do we reduce cognitive load of food dietization? Food dietization is the process of querying a user or a participant on a daily basis over the course of breakfast, lunch, and dinner. What did they eat today? How much did they eat? What was the caloric content of what they ate? Unfortunately, this has a large cognitive load on research participants because they get tired of answering questions or they forget. So we wanted to create an application that made it easier for participants to fill out this information. What we did was we monitored their location through a GPS continually, and if they stayed at or near a restaurant for longer than 15 minutes, we queried them and asked them to input the information on what they ate at that restaurant. And so in addition to a complete caloric breakdown for each participant, we also got information on restaurant type, visitation frequency. So in summary, we created a platform that is highly composable and that you can combine different sensor streams to guide interventions. It's expressible so that you can implement and test any trial design hypothesis. And it's also very simple to use so that a clinical researcher with no technical background can implement any arbitrary study. So I'd like to thank our shepherd and reviewers. This work was partially funded by NSF ERC Paths Up, as well as NSF Expeditions in Computing. And I'd like to thank my co-authors, Amrita Pai, Jian Kao, Nidal Mokadam, and Ashutosh Sabharwal. And if you have any other questions, you can ask me questions, but if you want to find out more, you can visit our website, which is at this QR code. Thank you.